Hi, my name is Tomasz Wilmanski, and I'm the lead author of the recently published meta-article, Heterogeneity in Statin Responses Explained by Variation in the Human Gut Microbiome. This research was performed at the Institute for Systems Biology. The study utilized samples from over 2,800 individuals. About 1,800 of those were from an American cohort, with a further validation in an independent European cohort. Approximately 20% of these individuals were on statins, one of the most commonly prescribed drugs in the Western world. Statins are a cholesterol-lowering medication, and while they're effective in decreasing cardiovascular disease risk, they come with certain side effects for a subset of individuals, including increased insulin resistance and increased risk of type 2 diabetes. We combined a series of multiomics measurements, including the genome, gut microbiome, and blood measurements, to identify how the gut microbiome may explain responses to statin medication. What we identified was that a Bacteroides dominant, alpha diversity depleted microbiome composition corresponded to a stronger statin on-target response, so a greater decrease in LDL cholesterol, but also greater risk of metabolic complications, as assessed by markers of insulin resistance. A rhinococcus dominant microbiome, on the other hand, still showed a response to statins, but virtually no risk of adverse effects. What was quite striking was that all these microbiome statin associations were independent of previously identified genetic variants predisposing you to a statin response, indicating that in the future we might combine genomics and gut microbiome analysis to better inform therapy for statin patients. However, the study is not without limitations, one of them being that this is a purely observational study, and further experimental work is needed to fully understand the complex interactions that might be taking place.